So we are going to pick up right where we left off last time, talking about the 5 I's of, the 5 I's of bacterial and microbial laboratory techniques. Okay, the 5 I's that are required for us to kind of study and look at microbial organisms. And I know it's hot in here. I had somebody come up and ask me if I could turn the heater down, and I can't. Believe me, I'm really wishing I had worn an undershirt underneath this really thick sweater. But I can't do anything about it. So it's just going to be hot in here. I'm sorry. But we are picking up with our next I. So our second one was incubation, which had to do with creating a temperature and gaseous atmospheric environment that is conducive to growing whatever bacteria we want. And that was our case study that we talked about. So now we're moving on to our third I, which is isolation. And isolation, just like anything else, any other time that we talk about isolation means separating things from each other, right? And that's what this one is as well. So isolation of bacterial organisms is just separating one species of microbe from another. And there's a lot of reasons why that might be important, but specifically for clinical samples, it's really important for us to separate what is causing an infection from what is not, right? Remember that there is bacteria growing on and in every surface of your body, just about. There are a couple that don't have any, but just about everything on and in your body has some kind of bacteria in it. So if we are testing you for, and we're going to talk about our case study in just a minute, if we're testing you for strep throat, we take a swab of your throat, there may be bacteria in there causing a problem, causing you to be sick, but there is going to be dozens of bacterial specimens in there that are not causing you any problems. They're part of your normal body. And our job, or my job as a medical laboratory scientist or a microbiologist, is to know what is causing the problem versus what is not. And when we are testing human samples for bacterial infection, it's really important that we have one single bacterial species to work with. That is something called a pure culture. And it's critically important to the rest of this process. So in order to do the last 2 I's, which we're going to talk about in a second, you have to have something called a pure culture. And a pure culture is kind of just what it sounds like. It is a culture that contains one bacterial species. Growing on media. Okay. So if you have a mixed bacterial culture, that's the opposite of a pure culture, is a mixed. If you have some kind of media or something else that's growing multiple types of bacteria, we are not going to be able to do the rest of this process. The identification and, I can't remember what word we use, identification and inspection, couldn't remember the I. Inspection and identification, in order to be able to do those two, you have to have a pure culture. So this step is critically important to the rest of the steps. And what a colony is, an important part of this process is that when you put bacteria on an agar plate and incubate it, so when you first spread bacteria all over a plate, right? And I know you guys haven't really done this yet in lab, but you'll just have to take my word for it at this point. Can you see any bacteria growing on any of the surfaces around you right now? No, they're invisible. That's the whole point. When you first put bacteria on an agar plate, when you first inoculate it, they're going to be completely invisible, okay? And that's what this is showing you, is that somewhere, right here, right after you inoculation, those bacteria are spread out all over the plate, ready to grow, but they're still invisible. We can't see them. But what's going to happen over however long we incubate this, whether it's 24 hours or 48 hours, is those bacteria are going to divide. And then they're going to divide again. And then they're going to divide again. They're going to divide again and again and again and again and again, hundreds and hundreds and hundreds of times. Most bacteria can divide about every 15 to 20 minutes. Okay, so think about how many times that will happen in a 24-hour period. And what you're going to be left with is millions and millions and millions of bacteria in one single spot, because we put them on a piece of media and they are going to be floating or sitting on one single spot. And what that is going to give us is visible colonies. So these invisible bacteria that get spread all over the plate, they sit in one spot and they divide millions of times. And you end up with millions, if not billions, of individual bacterial cells sitting in one single spot. And that is going to give us something that is visible. And this visible chunk or glob of bacteria is called a colony. Okay, it's the visible growth of organisms. Okay. And so what we'll be left with is something that looks like this. So you can see pretty clearly here, if you look at the color of these different colonies, I see two different colony types. Okay, and the reason I know that is because of the two different colors. This one right here is clear, white, whatever you want to call it, yellowy color. And this one is very clearly pink or red. All right, so there are two different bacteria growing here. They have two different characteristics. One is clear, one is red. So if I have this mixed bacterial culture, it is my job as a microbiologist to separate those two. And that's what isolation is. And subculturing, so what we have down here, isolation techniques and subculture, what subculture is it's taking a little piece of one colony of bacteria and putting it on a new plate. So in this case down here, if this is somebody's infection, somebody has a suspected urinary tract infection, and there are two different bacteria growing, we need pure cultures of both of those bacteria. So what I'm going to do as the microbiologist is subculture both of those organisms. So I'm going to take an inoculating loop, which is just a little sterile loop, and I'm going to pick up a little bit of this and put this on its own plate and streak it out again. So we'll call this one A. So we'll take a little bit of A, a little piece, we'll take one inoculating loop, take a little bit of A and put it onto its own plate. And then we're going to take a little bit of B, we'll circle in orange. So this will be B. We'll take a little bit of B and put B on its own plate. Now, what we have to do now is we just inoculated new media. So we got to go back to the beginning with that. What do we have to do after we inoculate something? What's step 2? What's the second I? Incubate, right? We inoculated it, so we spread it on some new media. It's invisible. There's not a lot of it there. Now we're going to stick it back in the incubator, and we're going to incubate it for another 24 hours. All right, and what we're going to be left with at the end of this is two pure cultures, a pure culture of A and a pure culture of B, that we can then do what we need to do with. Okay, so that's what subculture is. It's the process of separating organisms. All right, so to put that in the context of our case study, and I want to preface this by saying that this next part is not something that I expect you to understand how to do, okay? I don't expect you to be able to pick out what is a pathogen and what is not a pathogen, because it took me 5 years of study to figure out how to do that. I don't expect you to know how to do it in 10 weeks, okay? But the part that I'm going to tell you next, it's just to understand the steps. Okay, you don't have to know how to do this, and I'll explain in a minute. So we have our case study. We have this person who has a really sore throat and has a bunch of pus growing in the back of their throat. They possibly have strep throat. Okay, that is what the doctor figured out, that they probably have strep throat. So a nurse came in and took a swab of their throat and sent it down to the microbiology lab. The microbiology lab then inoculated media with that swab. They incubated it overnight, and this is what we have. So after we incubated it overnight at the correct concentrations and temperatures and all that, this is what we have. This is the original direct plate. So this is the plate, this is the piece of media that from that original swab, the original plate. And you can see here that there is a bunch of stuff. Probably at least 2, I see maybe 4 different types of organisms, which, like I said before, is not that surprising. Throats have lots of bacteria in them, okay? But a lot of that is normal. A lot of that is perfectly normal. Now, this is the part that I don't expect you to know how to do, but as a microbiologist, I know, looking at this plate, what is abnormal and what is causing this person's problem. What is causing this person's problem is this colony right here. And I only know that after years and years and years of experience. That's not something I expect you to know how to do. But what I expect you to know is that this original plate is very, very mixed. We can't do anything with this mixed plate. There are at least 4 different types of bacteria on here, probably more. So my job as a microbiologist is to look at this plate and say, okay, everything on here looks normal except for this one weird looking colony. Okay? And what I will do now is I will subculture that one colony. I will take just a little bit, I'll take an inoculating loop and take one little bit of that colony that I suspect is causing the problem. And I'm going to take that and I'm going to subculture it to a new plate. So I'm going to inoculate and incubate another plate. And then after another 24 hours, we will have a nice pure culture. And you can see, even if you don't really know what you're looking at, that this plate has bacteria of different sizes and color doing different things. They visually look different from each other, right? You can tell that there's multiple different things growing here. But when you look at our subculture plate, you can see that all of that looks very, very uniform. Everything looks the same. And that's how we know that we have a pure culture. All right? And that is what you have to have. This pure culture over here is what you have to have in order to do the next steps. Now, something I want to point out here is microbiology is not a fast process for the most part, because now this patient came to the doctor, say they came in on Monday. All right, Monday they took the swab, it got sent down to the microlab, it got inoculated and then incubated for 24 hours, okay? Now the microbiologist is looking at it on Tuesday. That is when the microbiologist sees this plate, is on Tuesday, that mixed plate. On Tuesday, they subculture the colony that they think is having the problem, and they put that on a new plate. Well, now we have to incubate that for 24 hours, okay? And that's when you get this colony over, this plate over here. Now it's Tuesday, okay? And the person still doesn't have answers. So that's something to think about as we go through all of this, is a lot of microbiology and a lot of, you guys are gonna be on the other side of this. You can't diagnose infections a lot of the time really quickly. Infections are gonna be slow to diagnose. They take at least 2 days, if not longer. So microbiology results, if you ever get frustrated when you're a nurse and you're calling down to the lab because you desperately need to find out about this thing and this thing and this thing, it's like, I can't make the bacteria grow any faster. It's gonna take 2 or 3 days. Okay, this takes a long time, which is an unfortunate part of the process. It's something that we don't like, and it's something that a lot of technology is trying to currently overcome. So that's something that you guys might get to experience during your careers, is new technology that makes this faster. Okay, so that is our third step, right, is isolation, separating bacteria from each other. Our fourth step is going to be inspection. And all inspection is looking at the bacteria. All right? It's just visually looking at the colonies. Now, visually looking at the colonies and bacteria can happen in 2 ways, right? Remember, macroscopic is macro means big. So this is just what you can see with your naked eyes. And a lot of this is going to have to do with what the bacteria looks like on the media. And this is something that you're going to talk way more about in lab, but this is things like color of the colonies, the size of the colonies, the texture of the colonies, the smell, all kinds of different things. But these are things that you can visually see with your own two eyes. You can see right here, this is a collection of a bunch of different plates. Every bacteria on there has different characteristics, right? Some of them are pink, some of them are red, some of them are green. Bacteria really grow that color. That green color is a natural color of specific bacteria. Red, pink, yellow. They grow in different sizes, right? You can visually see that like this colony right here is a lot bigger than this colony down here. Okay? And when you're looking at them in person, you can see that they have different textures. Some of them, and I know these are weird words to apply to bacteria, but some bacteria have like a creamy texture. Some are dry and crumbly. Some are wet and mucoid. Some are sticky. Okay? They're all going to have these different textures. And all of these things will give us clues as to what this bacteria is. Because remember, at the end of the day, with all of this, what we are trying to do is figure out what is making this person sick. And it's my job as a microbiologist to figure out what in this person's sample is making them ill. And is it a bacteria? And if it's a bacteria, what bacteria? And when I figure out the bacteria, it's important to figure out what antibiotics are going to kill that bacteria. But we'll get there later. Okay, so the macroscopic appearance is all going to lead us to a lot of clues about what the identification of this organism is. And another way of visually inspecting bacteria is by using a microscope. So this is using the microscopic appearance. Okay. And this is looking at bacteria underneath a microscope. And we're going to talk more about this in just a second. I have a bunch of slides on it in a minute. But certain species and genuses of organisms are going to grow under a microscope. They're going to look different ways. They're going to have different sizes. They're going to have different shapes. They will have different arrangements. We're going to talk all about this in a minute. But there are visual differences that you can see under a microscope that, again, will help you identify this organism. Okay, so again, this is our case study. We already saw the subculture plate, but this is our pure culture, our subculture plate. But this is what the bacteria looks like from this patient's throat culture. And again, this is not really the part that I expect you to know how to do. I'm just giving you examples. But there are a lot of things that I can tell from this picture. I can tell you that this bacteria is relatively small. It's kind of clear. It looks kind of wet. It's shiny. It's doing something called beta hemolysis, which you don't have to write down and we'll get to later. But it's doing something called beta hemolysis. They're pretty flat. They have smooth margins, which means the edge is smooth. Right? There's a lot of different things. That is what I can see visually. What I can see macroscopically, if I stained this and put it underneath a microscope, this is what I see under a microscope. And again, this tells me a lot of information. We're going to talk more about this in a minute. But I have a pretty good idea looking at these two things what this organism is. I have a really, really great guess just based on these things. Okay? So that is inspection. It's looking at the physical characteristics, both macroscopic and microscopic, of an organism to help identify it. And then our last step is something called identification. So identification of an organism, the definitive identification of an organism can be a really complicated process. At this point, with our case study, after we do visual inspection, I, as a microbiologist, will have a very good guess at what I think this bacteria is. But the truth is that a lot of bacteria look similar. Okay, a lot of them you're going to look at if you're in lab, you're going to start to look at these plates and be like, they all look the same, right? They're all white and they're all a little bit clear. They're all the same size. They mostly have smooth margins, right? There are some clues, but a lot of bacteria look the same. And then one of the other things I like to say about bacteria is that bacteria don't read textbooks, okay? So we have a bacteria that's supposed to do beta hemolysis, but half the time it doesn't. Or it's supposed to be mucoid, and sometimes it's dry, okay? So inspection can only get you so far. You have to do a definitive identification. And definitive identification is usually going to involve some kind of what we call biochemical or genetic testing. And this is a process that, again, if you're in lab, you will spend a significant amount of time learning what these things mean. We're not going to talk about it in here, but you are going to learn the different types of biochemical tests that separate Staphylococcus aureus from Staphylococcus epidermidis, and how you can put, E. coli in a lactose tube to figure out what species it is, right? All of these things are involved in the definitive identification. Okay. Now, for a long time, manual biochemical testing was what we did. It was basically techs would take dozens of different tubes, which you can kind of see up here. They would take dozens of tubes, and each one of these tubes tested for something specific. Okay. And I know these words don't mean anything to you, but we were testing for things like, does this organism have the urease enzyme? Does this organism use lactose as a food source? Does this organism use citrate as a food source? All of these different biochemical properties. And we would test every bacteria for dozens of different things. And after we did all of that testing, we would look it up in a manual, and there would be a manual that said, this organism is positive for this, negative for this, positive for this, positive for this, negative for this. And you'd have to line them all up and figure out which organism it was based on what it tested positive for. Okay, so that was manual biochemical testing. And that was done for decades. This takes a lot of training. If you're in the lab, you are going to be doing some of this. If you're in lab at the end of this quarter, a urease and a citrate and a lactose is not going to be like a foreign language to you. You are going to learn what that means, and you're going to learn how to interpret those results and what species of bacteria are positive and negative for those results. Okay? But for a long time, that was how this was done. And then something came in. These were strip testing. So manual biochemical testing was tube tests and strip tests. And strip tests are basically it was a way of taking all these dozens of tubes and condensing it down into something a little bit smaller. Okay? But it's basically the same idea. It's just on a much, much tinier scale. So put all of these tubes, and it made the test much, much smaller, and put these on these little really convenient strips. So you could just add all your bacteria to those little strips, and it was easier to read. Right? So that is kind of the, I don't know the word for it, but the vintage way of doing it. Nowadays, most identification is done through automated testing. And automated testing is the process of using instruments to perform biochemical testing. So instead of a microbiologist having to line up 17 tubes, add bacteria to each and every one of them, wait an appropriate amount of time, usually between 16 and 24 hours, and then manually recording the results and looking that up in a manual and figuring out what the identification is, we now have instruments that do all of that for us. So you put a little sample of the bacteria in a tube on these instruments, and in usually about 12 to 24 hours, they spit out an answer for you. But the idea is the same, it's just an instrument doing it for us. Okay, an instrument is doing all of these tests, looking at the color changes, comparing those color changes to a preset manual, and telling you what this organism is. Now, this is, again, the most common way that this is done now. Most hospitals and doctors' offices and any kind of identification is going to be done through these instruments. And these instruments are just shining achievements of medical engineering. They're amazing and they're very complicated, and they save us all a lot of time. Okay, and so this is what an example of an automated testing result would look like. For our case study, right, we had our subculture plate, and we had a good guess at what our bacteria was, but to definitively identify it, if we were working in a hospital, we would take a little sample of that bacteria, put it on an instrument, and 12 hours later, it would spit this out. And you can see at the bottom, these are, and I don't expect you to understand all of this, but those are all the biochemical tests that this instrument ran, okay? And it has all of these tests, and it tells you whether it's positive or negative for all of these different things, and it compares that to a database, and it tells you that your bacteria is Streptococcus pyogenes, which is the bacteria that is responsible for strep throat. So our patient has definitively strep throat. And that is what happens every day for hundreds and hundreds and hundreds of samples for people who have infections. When I worked at St. Francis, we would get, on average, between 100 and 150 new urine samples every day. Just urine samples. Okay, we'd usually get between 20 and 30 new wound cultures. We'd get between 30 and 50 respiratory cultures every single day. And St. Francis is not that big of a hospital. So they're, pretty medium-sized, I would say. Pretty medium-sized. And so that just kind of gives you the idea of the scope of work that a microbiologist has and what their job is, and gives you an insight into some of the lingo, right, the words, because I'm going to use the word colony and stuff like that a lot. We're going to talk about incubation a lot, and it's important that you know what those words mean. So that's the process of all of this. And again, it's important to remember at the end of the day that all of this kind of theoretical talking about bacteria, talking about what they look like on plates, it's, again, tied to a person. There is a person on the other side of those swabs that has a problem. They are sick and they are hurting, and it's our job to figure out why. Okay, it's not just looking at the weird colors, okay? It's important, and it's important work, and it's important to understand how it works. All right, so now we're going to spend a little bit of time talking about microscopy, which is a hugely important tool in microbiology. As you can imagine, organisms that are too small to be seen with the naked eye, being able to see them is an important tool. And the only reason we're able to accomplish that is by using microscopes. Now, I am going to skip a couple of these. Again, I spent too long talking about some stuff earlier. So there are a couple of slides that we're going to skip over. And again, I'll put X's through them, and you are not responsible for that material on the test. But like we talked about in the last chapter, the discovery of the microscope greatly impacted the study of microorganisms. Being able to see different organisms, classify them based on their size, their shape, other characteristics, gave scientists a much better understanding of microbiology. Being able to pinpoint, oh, this one is different than this one, and this one is different than this one, because of things that I can see. And you can see here, this is just some examples of bacteria that grow in different ways, okay? And they're very visually different. These little spiral guys down here, I would guess that those are probably syphilis. Syphilis likes to grow in little spiralies like that. And there's a lot of things that you can tell from just looking at a bacteria underneath a microscope. They grow in different sizes, they grow in different shapes, they grow in different what we call arrangements, which I'll talk about in a second. So visually, there's a lot that can be done just by looking at microorganisms underneath a microscope. And this is just a little bit of a reminder, but this is a chart that shows the relative size of many different cells. And just as a reference, the human eye can perceive down to a little less than 1 millimeter. Okay, so the human eye This is the range of the human eye. The human eye can perceive down to about 1 millimeter. Sometimes a little bit less, sometimes a little bit more, depending on the conditions, okay? Like right now, I know that there's a bunch of dust floating in the air, but you can't actually see it. But sometimes if the lighting is just right, the lighting hits it, like at sunset, the lighting hits it, and you can see all the things floating in the air, right? So sometimes if the conditions are just right, you can see just below 1 millimeter. But for the most of us, on an average day with average lighting, we see about 1 millimeter, which is about the size of a louse, which is like a lice, little tiny bugs, or mold spores. Okay. So this is most or some eukaryotic cells are about in this range. And most eukaryotic cells fall just below the 1 millimeter. So a lot of eukaryotic cells you can see if you look really, really closely. For example, a human egg is just shy of 1 millimeter, which is weird to think about. But you can actually, if a human egg is in a petri dish and you're looking at it with just the right light shining on it, you can see a human egg floating in solution. Okay? But most of our pathogenic organisms are going to fall into microscopic range. So the human eye, again, can see, this is the macroscopic view, can see at about 1 millimeter. Most eukaryotic pathogens fall around what's called 100 micrometers. Okay, and a micro is 1/1,000,000th of the size of a meter. So 1 micrometer is about 100 times smaller than 1 millimeter. So 1 millimeter to 1 micrometer is about 100 times smaller. That's not the best color, but 1 millimeter to 1 micrometer is about 100 times smaller. So most bacteria, most human bacteria, are going to be about 100 times smaller than what we can see with our naked human eyes. All right? And it just so happens that about 100 micrometers to 1 micrometer is the range of an average light microscope. Okay, so there's lots of different types of microscopes. The ones we are going to be talking about and the ones we are going to be using are light microscopes. They shine light and they use lenses to reflect the light, and that is how you see an organism that is smaller than you would be able to see normally. Okay, so this is kind of what we call a regular microscope. The microscopes that we use in labs here, they're probably the ones that you're very familiar with. I have a picture of one in a little while. But regular light microscopes can see eukaryotic organisms, like these guys. They can see human cells, and we'll look at an example in a minute. But they can, most importantly, see bacteria. Now, if you want to see something smaller than that, like most viruses, you actually need a special type of microscope. And that special type of microscope is called an electron microscope. You don't have to understand the difference or how they work, but it doesn't use actual visible light. It uses waves of electrons that are smaller than visible light. It has to do with wavelength and things bouncing off each other. You don't have to understand that, but what you do have to remember is that viruses are not visible with our light microscopes, okay? So you will never be able to see a virus in your regular lab class, okay? Because we use regular old light microscopes. If you want to see a virus, you have to have a special electron microscope, because viruses are about 100 times smaller than bacteria. All right, so this is what an average microscope looks like. We're not going to go into too many of the details as it relates to this class, again, we'll go into a lot more detail in lab. I know I keep saying that. But this is a picture of a standard light microscope. And it is what we are going to be focusing on in this class because it's the only ones that you're going to ever use. So we're not going to talk about electron microscopes because those are very complicated pieces of equipment that you're not going to use in this class. Maybe you'll get lucky and get to use one day, but not in this class. I don't think, sorry, I'm making choices about what we're covering here. All right, I think we're actually going to skip most of this. Okay. Just remember about the light microscope is what we're using in class, and that's about it. If you want to see viruses, you have to use an electron microscope. We're going to skip the rest of this. We're going to skip this too. Okay. I did want to show you this as just a point of comparison of what a light microscope can do. A light microscope allows you to look at things at very different levels of magnification. So it's how big you're making something. So the lowest magnification on a light microscope is right here. So this is the lowest, and this is the highest. And this is, all of these pictures are of human blood. So you know what human blood looks like when you look at it just macroscopically, right? It looks like a liquid. You can't see individual particles in it. It just looks like a liquid, right? When you put it underneath a microscope and you look at it under the lowest magnification, this is what it looks like. It kind of just looks like, it looks like purple stuff, right? You can't really see anything. You can see that there's individual little circles there, maybe, but that's kind of it. Okay, but when you zoom in a little bit, now you can see the individual cells. Those are red blood cells. And you can even start to pick out, this one is a white blood cell right there. You can see that it looks a little bit different. And then this is what it looks like under maximum magnification for a light microscope. You can see individual details. I could tell you what type of cell this is. I can see its nucleus, which we'll talk about later. But you can distinguish individual characteristics. Okay? So the point of this is just to show you the difference between the lowest magnification and the highest. Light microscopes are capable of a lot of very powerful things. But that is the difference in them. Okay, we're not going to talk about resolution. Like I said, we're skipping a bunch of stuff. We're not talking about this. This has to do with resolution. We're not talking about contrast. Skipping all of it. Okay, we are going to talk about this. Okay, so when we are looking at stuff underneath a microscope, right, it does require some amount of preparation. You can't just, I know that they sell those little things now where you like can just look through it, and they're like little microscopes that they sell from drop shipping companies that you can buy on Amazon. But for the most part, samples are going to require some kind of preparation. So we're going to take our sample, whatever it is, whatever we're trying to look at, and we have to do something to it in order to put it underneath the microscope to look at. We can't just shove an entire swab underneath a microscope. That's going to do nothing for us. Okay? And there's two major ways that we prepare specimens for looking at them under a microscope. And those two different ways are using a wet mount or a wet fixation or a wet preparation, and then what we call fixation and staining. And we're going to talk about the pros and cons of each. Okay, so a wet preparation is exactly what it sounds like. It's taking liquid sample and putting it on a glass slide and putting it underneath a microscope. Okay, and that's it. It's just taking a drop of liquid. A lot of times this will be in clinical samples. I know this is weird for a lot of people to think about. And some people always get weirded out the first time I say these words, but we're gonna get used to it, okay? So this could be something like vaginal fluid, right? Somebody who's having some kind of vaginal infection, there's a lot of fluid coming out of it. You can take a little sample of that liquid, put it directly on a microscope, and look at what's happening. You could take urine, pus from a wound, anything that's liquid, cerebral spinal fluid, which is fluid from around your brain. You take a little sample of that, put it underneath a microscope, and look at it directly. Now, the pros of this are that it's fast and easy. okay? It takes 12 seconds to put something on a microscope slide and put it under the microscope. You're literally just taking a drop of something and sticking it under there. There's basically no other prep work required. There's no staining, there's no wait time. It's very fast, and it's very easy. And the other pro to this, the other good thing, is that it allows you to see cell movement. Because when we put a liquid sample that we haven't done anything to underneath a slide, those organisms will still be alive. So if we are looking for something that moves, that is going to be our best bet. There's a specific type of, which we'll talk more about later, but there's a specific type of infection that happens in vaginal tracts, and you don't have to write down the name. We'll talk more about it later, but I'm just telling you stories. It's called trichomonas. Trichomonas causes trichomoniasis, which is a parasitic infection that happens specifically in vaginal tracts. And trichomonas literally swims. So when you take a specimen of vaginal fluid from somebody who has trichomoniasis, you put it under a microscope and you can see those suckers swimming around underneath the microscope. And yes, it's gross, but it's also kind of fun when you're the one that sees it from the lab and you're not experiencing it. I'm sure it's not fun to experience. But seeing that movement is key to the diagnosis of trichomoniasis. And if you see something moving like that in a vaginal sample, you can diagnose somebody with trichomoniasis that took a minute, literally. They sent it down from the ER. Somebody's got vaginal issues. They send that down from the ER. You pop it on a microscope slide, look at it. You see them moving around, done. Diagnosis made. That's it. We don't have to incubate. We don't have to do anything else. So if we want to see movement or live organisms, a wet prep is the way to go. Now, the cons of this is that there is less detail. Specifically because we're not staining anything. And it is not useful bacteria. It's not useful for bacteria. And the reason for that is because you actually can't see as much detail on a wet preparation to be able to visualize bacteria. Bacteria are just gonna look like little dots, okay? So a good example of that is this right here. So this is a wet preparation of a vaginal sample. Okay, so this is a vaginal sample from somebody in a hospital, I don't know who, but this is what it looks like underneath a microscope. Now notice that nothing in this slide is stained, nothing has any kind of color, there's no contrast, everything is just clear. And if you don't know what you're looking for, it can be really difficult to see things, okay? And there are some important things that can be seen. This is yeast right here, which is likely what's causing this person's problem. They have a yeast infection in their vaginal tract. So you can see yeast, you can pick them out because they're bigger. But you see all these little black dots in the background? That's bacteria. Now, can you tell anything about those bacteria? No, they look like specks of dust. I can't tell anything about that. But to look at it from another way, so if this person had a bacterial infection, a wet prep would tell me nothing and it would be useless. But if this person has a yeast infection, yeasts are eukaryotes, which we'll talk more about later, yeasts or anything that's moving, like trichomoniasis, a wet prep is gonna be great. And it's fast and easier than doing anything else. All right, so that's the pros and cons of a wet prep. Now, a fixation technique is the process of taking a bit of sample and usually using heat or chemicals to stick that bacteria to a slide. And the reason that's important, you'll go into in lab, it has to do with using immersion oil, but you basically dry out the specimen and you fix it, which means you stick it. Fixing means making sure it's stuck. Okay, it's like fixing wallpaper or something. You stick it to the slide and then you put chemicals over it to stain it different colors. Now, the pros of this is that you can see more detail. It's just kind of the opposite of the wet prep. So you can see much more detail. This is good for bacteria. And because you're staining things, which we're going to talk more about in just a second, you can stain things different colors. For example, this is an example of things that have been stained different colors. And this will tell us whatever we're trying to find out based on what color the things turn. So if we need to know something about specific details of an organism, that is what we want to use, okay, is a fixation. It shows more detail. It can see bacteria down to individual cells, and it can be stained different colors. It's helpful. The cons of this, the first one is that there will be no movement, because when you fry something over an open flame or dunk it in chemicals, it's going to die. The cells in there are going to be dead now, so they are not going to move. So if you are looking for trichomoniasis, this is not a good way to find it because it's going to be dead. You're not going to be able to see it. Okay, so there's going to be no movement, and it is more labor intensive. It requires much more work, not a significant amount of work, but it requires more work, takes more time, heat fixing and staining. There's usually wait times in between. Okay, it's a little bit more labor intensive and it takes more time. So those are our two major types of sample preparation. Wet preps are usually used for anytime you're looking for movement or you're looking for eukaryotic cells, bigger cells. Staining and fixing is usually used for bacteria and when you don't need to see movement. Okay? So they both serve a purpose. They're both very useful. It just depends on what you're trying to accomplish, which one you're going to pick. Okay? And so we are briefly going to be talking about what is called gram staining. And gram staining is by far, not even close, the most common stain in a microlab. Now, there are lots of different stains that can be used depending on what you're trying to see. If you're trying to see if something has endospores, you can use an endospore stain. If you're trying to see if something is tuberculosis, you can use a tuberculosis stain. There's lots and lots of different things that can be used, and we'll talk more about a lot of them later. But by far, the most common stain used in a microlab is a gram stain. And it differentiates organisms based on their cell wall. And I know a lot of these words don't make sense right this second. We're going to talk much more about this later. But what you have to remember for now is that gram-positive organisms are going to appear purple when you look at them underneath a microscope. And gram-negative organisms will appear pink. Okay? So is this gram-positive or gram-negative, this guy right here? That's positive, right? It's purple. And then this one is negative. Some people think that looks red. You can call it red or pink or whatever you want, but it's not dark purple. Okay, so when you look at them in comparison, this is purple. Some people get confused because they're like, well, that looks like magenta to me, and magenta is a type of purple. When I'm talking about purple, I'm talking about purple so dark it's almost black sometimes, like a bluish purple. Okay? And we'll talk about more about what makes an organism positive or negative in just a little while. I think it's in chapter 3. But I think it's important to talk about here how important and critical a gram stain can be in the process of identification. Because the vast majority of bacteria that we are going to talk about throughout this class are either gram-positive or gram-negative. Okay? It's about 50-50. Half of them are positive, half of them are negative. So I want you to think about if somebody comes into the emergency room and they have a life-threatening infection. Their caregivers are looking at them, they have a high fever, their blood pressure is dropping, their heart rate is really high, they're sweating, they're delirious. They probably have a very serious infection that needs to be treated 24 hours ago, okay? But different bacteria respond to different antibiotics. Sure, there are antibiotics that you can give that cover most things, but the more information you have, the better chance you have at treating somebody. okay? So to give an example of this, I'm gonna give you, it's always just easiest for me to talk about it this way, because there was a time about 5 years ago that I was that patient. So I was in a car accident in January of 2020, and I had lots and lots of injuries. I broke both my legs, I broke my elbow, I broke my pelvis, I broke my spine, I broke all kinds of stuff. But the worst thing that happened to me was I ended up with a horrific infection because of my seatbelt, which probably saved my life and made sure I didn't die. But a little tiny cut on my lower pelvic region got infected. Okay, and I ended up with a huge infection that went from my hip bone past my knee. My whole thigh was completely full of bacteria. I had pus coming out of it. It was disgusting. So I went to the ER. I had just gotten sent home from the hospital. I had had 4 surgeries, I think, to cement my pelvis back together. And then they sent me home. And I developed this infection, and I went to the ER, and that is what my doctor saw when he looked at me. He said, "You have pus coming out of your leg. Your blood pressure is in the tank. Your heart's beating too fast. You're not getting enough blood. You have a horrific fever. You can barely talk. You are on the edge of sepsis and complete organ failure. And we need to treat you right now." And this is not a abnormal thing. Okay, people who work in emergency rooms see this all the time. It is a very common scenario. Now, all that other stuff that we talked about identification and all of that fun stuff. What was one of the things I said about it? Said it takes days, right? Bacterial identification, a full definitive identification, takes days. Do you think that I had time for somebody to figure out exactly what was growing in me before I died? No, I didn't. I didn't have 3 days to wait. They needed to treat me now. But what they can do to help direct treatment is a Gram stain. So Gram stains, when done correctly and promptly, take about 10-15 minutes. Okay, you can send a sample down to the micro lab, they'll stain it, and you can know in 10 or 15 minutes is what is growing in that person Gram positive or Gram negative. And now you've just taken all of the possibilities and cut it in half. So of all the bacteria that could be growing inside my body, there's now half. Okay? And that severely decreases the choices that need to be made. It gives the doctor a place to go. It gives them a treatment plan that they can start. All of that identification and stuff is still going to keep happening, and they will adjust as you go. But a Gram stain can be the difference between life and death. Not to sound too dramatic about it, but it can be, and it's important. Yeah, and when this happened to me, that's exactly what they did. They took a swab, they stuck it in the wound, they collected a bunch of the pus, they sent it down to the micro lab, and I actually went to St. Francis where I was working at the time, so my friends were texting me as they were looking at my slide from all of my infection, but I had a Gram negative bacteria. And so the antibiotics that they meant to put me on, you know, they have all of these choices about, okay, well, what do we want to do to stop her from going septic, from making this worse, all of these things. Well, now all the choices are cut in half because we know that this organism is Gram negative. So that's a place to start. And a lot of times with infections, things happen really, really quickly. And so, yeah, it's really hard to overstate the importance of a Gram stain. And sometimes I think it's important to keep in mind that all the technologies and all of the things that are available to all of us, sometimes the simplest thing, using a microscope slide and looking at it for 10 minutes is the best and fastest thing you can do. And that's going to apply to a lot of different things in your nursing career. You know, there's a lot of great technologies. I'm not saying we should get rid of all of them, but sometimes relying on your instincts and doing the simple thing is going to be the best one. Okay? And I will talk about my accident a lot more, just because I don't have to follow HIPAA laws. It happened to me, so I can talk about all the details, right? I have lots of stories about patients and things like that, but because of HIPAA laws, I can't talk about it. But because it's me, I can tell you all of my experience. And I think it's an interesting way to look at both sides, right? Like, I'm a microbiologist. I understand this stuff in theory. And I was on the other side of that for a long time. but I had an experience where I was put on the other side of caregiving, which I think gives me a unique perspective. So it's something that I'll talk about a lot. If you have any burning, everybody's always curious. And I will talk, I don't mind talking about it. So if you have any questions about it, you can ask me after class. But yeah, okay, so that's the end of chapter 2. We are gonna move on to chapter 3. I'm sorry, we have 15 minutes left, so we still got time. But does anybody have any questions about chapter 2 before we move on? All right. Okay, so chapter 3, we are going to begin looking at specific characteristics of bacteria and archaea. Now, and these are both specifically prokaryotes. Remember, we have 2 classes of organisms. We have prokaryotes and we have eukaryotes. We're going to talk about a lot of the differences between them and how we tell the difference between prokaryotes and eukaryotes. And it is important to remember as we go through this that there are pathogenic microorganisms, so microorganisms that cause disease in all of these groups. Okay, prokaryotes and eukaryotes can be infectious pathogens. A lot of times people think microbiology and they think bacteria, and that's a lot of it, but it's not all of it. Okay. So again, in this chapter, we're going to be talking about the basic biology of prokaryotic cells. So the things that they have in them that make them act certain ways, what characteristics they have that allow them to cause infections, what characteristics they have that make them different than human cells or eukaryotic cells. We're going to be talking about all of this. Keep in mind that as we're talking about this, for the most part, we're going to be discussing bacterial cells. And the reason for that, we're going to be talking mostly about bacterial cells. The other half of prokaryotes is archaea, right? Talked about it over here. Bacteria and archaea are both prokaryotes. But as of right now, as far as I know, and I think as far as everybody else knows, there are no archaea that cause human disease. So archaea are part of the environment. They can be really important in the environment, but I don't know of a single one, I don't think anybody else does either, that causes human disease. So since this class focuses on human disease and how organisms affect human health, we're mostly going to be skipping archaea. But know that they're there. They are prokaryotes, and we will have a brief discussion at the end of this chapter about how prokaryotes, bacteria and archaea are similar and different. But as I go through these slides, I am mostly talking about bacteria. All right, so just as a review, we talked about this in the last chapter, or the last lecture, I think. All cells, including prokaryotes and eukaryotes, this is a little reminder, have 4 things in common. All cells, in order to be a cell, has to have DNA, or genetic material, ribosomes, which make proteins. It's a quick overview. Cytoplasm, which is the jelly-like substance that makes up the inside of a cell. And then the cell membrane, which is the boundary. All cells, prokaryotes and eukaryotes, are all going to have those 4 things. And I know I said this before, that there are not, one of my favorite little sayings is that there are very few absolutes in biology, right? There are very few things that are always true or never true. There's almost always an exception. This is one of those times that there's no exception. All cells, anything that we call a cell, has to have those 4 things. But what we're going to spend the next couple of chapters talking about is what these cells have in different, not in common, but not in common, how they differ from each other. Okay? And so briefly I want to talk about now the big differences between prokaryotes and eukaryotes. So prokaryotic cells, again, all things they have in common, they all have ribosomes, plasma membrane, cytoplasm, and then DNA, which is not on this diagram. The biggest difference and the most identifiable difference between prokaryotic and eukaryotic cells is that eukaryotic cells have what we call a true nucleus. And what a true nucleus is, and you can see it kind of labeled down here on this eukaryotic cell, this nucleus is where the DNA is, and it is surrounded by a membrane. So eukaryotic cells have a plasma membrane or a cell membrane, which is the outer boundary of what makes the cell, right? It's around the outside. It's like the eggshell. It's the outer part. Inside of every eukaryotic cell, there is a separate membrane. It's called the nuclear membrane, where the DNA is. Okay, it's a separate little room inside of a eukaryotic cell where the DNA is kept. Okay, so the DNA is kept inside of its own little room. It's surrounded by its own lipid bilayer, which we'll talk about later. But it has a separate membrane, which is a true nucleus. It is separated from the rest of the cell. Prokaryotes do not. Prokaryotes do not have a true nucleus. So that is the biggest difference between prokaryotes and eukaryotes. And I'm going to say this approximately 1,000 more times, and it's going to haunt your nightmares until the end of time, that eukaryotes have a true nucleus. Prokaryotes don't. That is the biggest difference between all of these organisms. Okay? And there are a couple of other smaller differences that we'll talk about as we go through. Eukaryotes also have what are called organelles. So eukaryotes have something called organelles, which we'll talk more about when we get to the eukaryotic chapter. Prokaryotes don't have them. And the way I like to think about these different cells is a eukaryotic cell is like an old house. I don't know why this is the thing that comes to my mind, but it's like an old house where every room in that house has a separate door. And to get to every, you go into the dining room and that's got a whole separate door. And to get into the kitchen, you got to go through a hallway and back into a separate door. And then every bedroom has its own door. Everything is separated. Everything has its own little compartments. That's how eukaryotic cells are. You have the one big membrane on the outside, and then all the rooms inside that cell are also separated by membranes. You have the nucleus and the organelles. They're their own little separate rooms where specific things happen. A prokaryotic cell is more like a newer house where everything is open concept, right? You walk into my house, I have a newer house. You walk into my house and you can see the kitchen, the living room, the dining room, all in one room, right? And I don't even really have hallways. And you walk into my master bedroom, which is not a thing I love, but it is the way it is. You walk into my master bedroom, you can see the bathroom, you can see the bathtub, the shower, and my bed all in one go, right? Everything is open. Everybody likes the open spaces in these new houses. And that is how prokaryotic cells are. Prokaryotic cells do not have separate compartments. They are all open. All right, that's the biggest difference. And again, this is just a reminder, we are going to be mostly talking about bacteria, but it is important to remember that both bacteria and archaea are prokaryotes. And there are lots of important organisms that fall into that archaea category. They just don't cause human disease, so they're not going to be our focus. So we're talking about these kind of two categories here, but mostly we're talking about bacteria. So when we're talking about bacteria, their shape and their arrangement of their individual cells is really important. A lot of the times when we're talking about bacteria, one of the first things we say about it is its shape and arrangement. It helps us classify the organisms into specific groups. And this will make more sense kind of as we go, but I wanted to introduce these terms now so we can talk about it. So bacterial shape, as you can probably guess, is the shape of an individual cell. Okay. So the major shapes of bacteria, and the kind of cool thing about bacteria is bacteria really only come in a set number of shapes. Okay. There's probably like 5 different shapes that bacteria kind of come in. And so we can classify an organism based on its shape pretty easily. The 3 major ones, and the only ones that you have to know for this class, are cocci, or I guess the individual. A single bacteria is called a coccus. Plural is cocci. Coccus, bacillus, and spiral. Okay, and this, you can see these over here. So a cocci is a single coccus, plural cocci, is spherical. It's a round shape. Okay, so cocci is round. Bacilli is plural, bacillus is singular, is what we call rod-shaped. And then spirals, as you can probably guess, are spiral-shaped. Some of them can be what we call vibrio, which is kind of a comma shape, but they are, again, the only 3 that you have to remember are coccus, bacillus, and spiral. So cocci or coccus is circular or spherical, bacillus is rod-shaped or elongated, and spirals are spirals. It means they can either be curvy, sometimes they're like corkscrews, sometimes they're just curvy, but they form some kind of spiral. Okay? So shape is the shape of an individual cell. And an arrangement are the way multiple bacteria or multiple organisms interact with each other. Okay. So it is how they interact with each other. It's how they form, I'm trying to think of the best word. It's how they form groups. And sometimes bacteria don't have an arrangement. Sometimes they don't like having friends, and they're by themselves, and they don't have an arrangement. Okay? But in the most common arrangements, you can see here. And a lot of it you can kind of tell by the prefix. So a diplo, what does di mean? 2. Di means 2. So a diplo is when 2 bacteria are often stuck together. Now you can have a diplococcus, which is 2 cocci stuck together. So notice that we're using the shape and the arrangement together. A diplococcus is 2 cocci stuck together. You can also have a diplobacilli, which is 2 rods stuck together. Okay. So diplo means 2. You can also have tetrads. What does tetra mean? 4. Tetra means 4. So a tetrad is 4 bacteria stuck together. Okay. So you get a little 4 groups. And you usually only see tetrads in cocci. You don't really have tetrads over here, but a tetrad is another one. Another type of arrangement is this staff arrangement. We call it a staff arrangement, and it's basically grape-like clusters. Okay, so it's just when the bacteria clusters together like bunches of grapes. Okay, so a staphylococcus are circular bacteria, cocci-shaped bacteria that are all stuck together like little grapes. And you'll probably recognize that name. Staphylococcus is a name that we have given bacteria that look like that. So Staphylococcus aureus was named that because its shape and arrangement is Staphylococcus. So that should tell you how important the shape and the arrangement of the bacteria is, if they've named it after it, right? That's an important thing. Okay, and then our last one that you have to know is strep. And strep means many bacteria forming a straight chain. So strep, the word we kind of associate with that is chain. So staph, we talk about clusters. Strep, we talk about chain. Okay, so you can have a streptococci, which is where you have lots of cocci stuck together in a chain. And then streptobacilli is when lots of bacilluses are stuck together in a chain. Okay, so those are the four that you have to remember: diplo, tetrad, staph, and strep. Those are the four arrangements you have to know. The three shapes you have to know are cocci, bacilli, and spiral. Okay. And this is just some pictures of all of these different types of bacterial shapes and arrangements. This right here is circular bacteria in pairs, right? So what would that be? Diplococci, circular bacteria in pairs. Okay, this is a tetrad, right? Cocci tetrads. As you can see, the four little bacteria. You see the separate little groups of four. Okay, this is a grape-like cluster. That's a staphylococcus. Okay, this is a streptococcus. Notice you can see the individual circles all stuck together in a chain. Oops. This is a bacillus down here. There's a streptobacillus. There's a little spiral. And then there's this is also considered a spiral, but this is specifically a Vibrio. It's like a comma shape. Right? Now, I only bring that up right now because I'm going to be using those words. I'm going to say words like staph arrangement and strep arrangement, and you have to know what that means. Okay? You don't necessarily have to know how to identify it for this class. For, again, lab, you are going to have to know how to identify that. But for this class, you just have to know what those words mean. Right? So this is the overall structure of a bacterial cell. We'll just do this slide, and then we'll be done. So this is the overall structure of a bacterial cell. Notice that the four components in the top left are components that all cells have, not just bacteria. So it is in all bacteria, but these four are also in all cells. But these four things we're going to talk about: cytoplasmic membranes, bacterial chromosomes, ribosomes, and cytoplasm. Those are the four things that all cells have, including all bacterial cells, because bacterial cells are cells. And then the rest of this are going to be in some bacteria. So we're going to go through a lot of these individually. We're going to talk about what their purposes are, what the structures look like in different types of bacterial cells. But just kind of keep this in mind as we go through each of these individual things, that on this overall picture, there are some things that all bacteria are going to have. We're going to talk about things that most bacteria have, and then we're going to talk about things that only some bacteria have. I do want to point out specifically a cell wall. The cell wall is a structure that the vast majority of bacteria have. Okay, so not all, we can't say that all bacterial cells have cell walls, but most of them do. And so cell walls are going to be really, really important. We'll talk about them. There's very few bacteria that don't have a cell wall. They are important too, but so yeah, that's the overview of a bacterial cell. And then we're going to spend the next couple of days talking about individual structures, again, what they do, what their purposes are, all that fun stuff. Okay?